So our next speaker is Meaghan McGeary, and she'll be talking about the evolution of the Sex Peptide Network, lineage-specific adaptive evolution, and gene duplication.
Hello. Thank you so much for having me today to talk about my undergraduate thesis work at College of the Holy Cross in Jess Finlay's lab. We've been interested in studying molecular changes that underlie phenotypic changes, especially in the reproductive system, and the Sex Peptide Network is a great model to study this. So a number of important changes occur in female flies after mating. These include female flies become unreceptive to courtship, increase egg production, ovulation, and egg laying, as well as increase sperm storage and release in the female sperm storage organs in the female fly. And these phenotypes can be seen for up to a week following mating. These are modulated by an interaction between sex peptide and sex peptide receptor. So sex peptide is a protein that is transferred from the male to the female along with sperm in the seminal fluid. Once in the female reproductive tract, it attaches to sperm tails and is stored along with sperm in the female sperm storage organs. Over time, after mating, sex peptide is released from the sperm tails and sperm storage and moves to interact with sex peptide receptor, its receptor in the female reproductive system. What we've just seen is the binding of sex peptide to the sperm tails and transfer and localization into the seminal receptacle. However, in order for these things to occur, a whole cast of characters is necessary. And so CG9997, CG1652, and CG1656 are three also transferred seminal fluid proteins from the male to the female that localize to the seminal receptacle along with sex peptide and the sperm. In order for these to reach the seminal receptacle after transfer to the male, and even for them to become transferred to the female, CG9997 must interact interdependently with CG1652 and CG1656, along with two other proteins, Antares and Aquarius, which are required for this as well. Intrepid is also a required network member that is important, but we don't know exactly where in the network it's required yet. Additionally, Seminase and CG17575 are proteins in the network that are required to localize sex peptide and sperm, as well as the other stored proteins into the seminal receptacle. Finally, once all of this has happened, three female proteins, Hadley, Fromoro, and ESP, must be present in order to support the interaction between sex peptide and sex peptide receptor in the female. The complexity of this network and the fact that these members all interact with each other, as I've just described, suggests that they may have co-evolved or evolved in similar patterns to each other. And in fact, several of the network members were identified based upon their coevolution with known network members. And so the complexity of this network is interesting, and we wondered how it evolved over time. And what I've just described is in Drosophila melanogaster. However, there are a number of important differences in Drosophila reproduction in many of the other Drosophila species. And so we were interested in understanding how these reproductive traits have changed over time. So we looked, in looking through the literature, we noticed some changes in sperm size. For example, along this branch here, we see longer sperm in Drosophila ananaceae and Drosophila bipectinata. And this could be important in correlation with the sex peptide network because we know that sex peptide binds to sperm tails. And so a change in sperm size could be correlated with a change in network proteins. We next looked at sperm storage. Since we know that sex peptide network proteins are important for successful and efficient sperm storage in the female sperm storage organs, and we noticed a couple of important changes along this line as well. Next, we looked at remating changes, since we know that sex peptide network proteins are important in modulating remating in the female after mating. And so we noticed that on this branch here, there's actually a remating less than daily change. So after this point, flies mate less than daily after the first mating. And finally, we looked for, we know of two important changes in gene expression. So firstly, as you can see in this paper, in this image here, Suda et al. Showed that sex peptide receptor began being expressed in the female reproductive tract after this point, after this branch within the melanogaster group. And so as you can see in this image, fluorescent sex peptide and sex peptide receptor can both be seen in the female reproductive tract. However, this is not seen outside of the melanogaster group prior to this branch. Further, we know that sex peptide does, this interaction between sex peptide and sex peptide receptor do not modulate receptivity changes, decreased receptivity in flies prior to this branch point. So this important change in gene expression may have an effect on changes in sex peptide network proteins. We also know of a gene duplication event of seminase, which is a sex peptide network member that occurred along this branch as well. And so given all of these reproductive traits have changed over time, as I've just described, we wondered how the sex peptide network has evolved over time and over the course of the Drosophila phylogeny. And specifically, how did these proteins come to work together, and how did they come to function in the network pattern that I've just described? One of the first things that we looked at is which sex peptide network proteins are present in which Drosophila species. And in this table, check marks represent presence of an ortholog. And of course, Drosophila melanogaster has the full set of sex peptide network proteins. And as you can see by the prevalence of check marks, almost every species has almost every sex peptide network, an ortholog for almost every sex peptide network gene, even including the species prior to the appearance of sex peptide receptor in the female reproductive tract. And those species with sex peptide receptor in the female reproductive tract are represented by species above the pink line. And so given that these these genes are so prevalent in these species, it implies possible conservation of these network proteins even prior to the appearance of sex peptide receptor in the female reproductive tract. And so we used PAML to test sex peptide network proteins for positive selection. DN-DS ratio is a metric that we used, and it represents a rate of non-synonymous changes over rate of synonymous changes in a gene. In most proteins, we see purifying selection and conservation of the gene. And so this is represented by a DN-DS ratio of less than 1. However, often in reproductive proteins, we see positive selection and adaptive evolution of these proteins, which is represented by a DN-DS ratio of greater than 1. We use two different tests. First, the CITES test, which allows us to test for recurrent positive selection across the entire phylogeny. And the way this works is it maintains a constant DN-DS ratio across the entire phylogeny to allow for DN-DS ratio changes in each site of the protein. Next, we use the branch CITES test, which tests for bursts of positive selection on a lineage that we specify based upon an a priori hypothesis in comparison to the rest of the background branches on the phylogeny. And again, it allows for a different DN-DS ratio across the different sites in the sequence while keeping the ratio constant on the foreground lineage and the background lineages each. And we tested specifically the CITES test within the melanogaster group because synonymous site saturation prevented us from good analysis on the rest of the phylogeny. So looking at the CITES test, there's two comparisons that we made. The first is the M7 versus M8 comparison, in which we compare a model that allows for positive selection against a model that allows for only purifying selection. And significance in this comparison indicates that the protein has a class of sites that are evolving under adaptive or neutral evolution. The other comparison is the M8A versus M8 comparison, which compares a model allowing for positive selection against a model allowing for neutral evolution. And significance in this test indicates that there is a class of sites in the protein evolving under positive selection. And so as you can see, CG9997, Fra-Moro, and Hadley are the 3 sex peptide network genes that are evolving under positive selection based upon their significance in both tests. Antares, CG17575, and Intrepid are evolving under neutral evolution based upon their significance in the M7 versus M8 comparison, but not in the M8A versus M8 comparison. And finally, the rest of the sex peptide network proteins are evolving under purifying selection as they were significant for neither test. And so as you can see, the majority of the genes in the sex peptide network are evolving under purifying or neutral evolution, suggesting conservation of these network proteins over time. Normally, or often, we see positive selection in reproductive proteins, which is different from other classes of proteins. However, here we see relatively slow evolution. And this may be due to a conservation of sex peptide network proteins and an important function in reproduction across the phylogeny as we tested. CG9997 may be evolving adaptively based upon an important function in transfer or its interdependency with the other sex peptide network proteins, even prior to the transfer to the female. Hadley and Fra-Moro may be important and evolving adaptively based upon their function in possibly the female post-mating response. Next, we tested using the branch sites test, as I alluded to, and we first tested up along this branch based upon the less than daily remating phenotype that I discussed earlier, as well as the appearance of sex peptide receptor in the female reproductive tract and the disseminase gene duplication event. A significant comparison between the alternative and null models here indicates that there is a class of sites evolving adaptively on this particular lineage. And as you can see, CG1652 is significant for this, as well as CG9997, ESP, Hadley, and Fra-Moro. We next tested along this branch based upon the bigger sperm that are seen in these species. Here, as you can see, 9 of the 12 proteins are significant and evolving adaptively on this branch. And so to put this all together, sex peptide network proteins may have been important and a conserved function in interacting together along the evolution in some conserved function, either in sperm transfer from the female to the male, possible protection of the female post-mating, or even in attaching something to sperm. Sex peptide receptor began to be expressed in the female reproductive tract along this branch, which placed an adaptive pressure on having more sex peptide transferred on sperm tails to the female, and inducing adaptive evolution in several sex peptide network proteins along this branch. In addition, the longer sperm, as you can see in the diagram here and on this branch here, induced adaptive evolution in several sex peptide network proteins along that branch. Further, the recurrent adaptive evolution seen within the melanogaster group may be as a result of the appearance of sex peptide receptor in the reproductive tract. CG9997, Fra-Moro, and Hadley may be evolving adaptively due to their important function in the sex peptide network. In CG997's case, in a transfer or dependency with other sex peptide network proteins, and in Fra-Moro and Hadley's case, in their important function in the female post-mating response. And so overall, we see conservation of sex peptide network proteins along the Drosophila phylogeny, accompanied with adaptive evolution that's lineage-specific, that may correlate with important phenotypic changes in the Drosophila reproductive system. And together, this paints a dynamic picture of protein evolution within the Drosophila reproductive system. I'd like to thank my fellow members of the Finlay Lab at Holy Cross for their support and advice, as well as the members of the Mandu Lab at Holy Cross, who also were very supportive and great with their advice, as well as a number of other people at Holy Cross. And I'd like to invite you to check out my colleagues' posters here at TAGC. Thank you, and I'd be happy to take questions.
We have time for one really fast question. Okay, everyone is waiting to ask you a longer question. Thank you, another really impressive undergraduate that we wish we all had. Great job.
Thank you.